Thank you very much. Nice to meet everybody, and please do feel free to ask any questions if you have. I'm going to turn off my video for now, and I'll bring it back later when we have our question and answer. Thank you again for joining. So for today's presentation, what I'm going to talk about are some examples of automated imaging applications for 2D and 3D assays. This is using the Molecular Devices Image Express family of automated imaging solutions. I'm really going to focus more on the applications. I will tell you a little bit about the instruments, but we want to focus more on giving you some example applications so you can see how you might take your manual microscopy applications and automate those. So thank you again for joining. So this is the typical way that many people are doing imaging. This is what I used to do myself, where, sitting at the microscope and focusing for your imaging assays, it can take a lot of time. I always like to tell a story. Basically, I feel like when I was a postdoc, I think I spent about 3 years in a dark room myself. So I know that this is very time intensive. With the automated imaging solutions that I'm going to tell you about from Molecular Devices, you can work with many different kinds of sample formats, so microscope slides, multi-well plates of various formats, formats that are optimized for both 2D or 3D type of assays, place into the instrument, few clicks of the mouse, walk away, images are automatically focused, images are acquired, saved, and data analysis. So at the end, you can come back and you can have those results. Now, this can save a lot of time, but it is also very enabling for being able to look at more conditions, more drug concentrations, more compounds, and so on. So it's about, being able to save you time, but also enabling to get more data and look at more conditions. Now, the workflow in an automated imaging or high-content screening is, of course, to be able to acquire those images in an automated way with the focusing across various formats. Having image analysis tools, this is an example of a neurite outgrowth, where we're finding the cell bodies and the associated outgrowths. Being able to output that data or access that data on a summary level for the whole image, as well as cell by cell. Each row here is an individual cell measurement, for example. And then when you're moving into automated imaging, the last step in the workflow is really how to make sense of the data. You will have a lot of images, you'll have a lot of data, so numbers, and really how to make sense of that. We call that data mining, or this could be data visualization, depending on the level of throughput that you need. Now, at Molecular Devices, we have a family, in our Image Express family of imaging solutions. And as I mentioned, I'm going to focus less on each, the individual specifications, but I just wanted to start by introducing the family where we have wide field imagers, we have confocal imagers. This is spinning disk confocal, so for all the platforms you can work with live cells. We have laser-based systems, we have LED-based systems. So we have a large family of different solutions, and that's both on the hardware and the software side. So let's jump into looking at some of the different applications. Now, in automated imaging, these applications can be quite varied. So you can think about what you're doing on a microscope now and automating that process, acquisition through data mining, as I mentioned. And really, we cover hundreds of applications from A to Z. Here are some examples, some key applications, such as looking at cell proliferation, looking at apoptosis, autophagy, DNA damage. These are just a few examples here, looking at assays, doing co-culture type of assays, migration. Don't worry, I know that we have a 45 minutes or so together here today. I won't go into all of these applications, but just wanted to give you a little bit of a highlight of the types of applications that our customers are using our Image Express systems for. And then from M to Z, you see examples of looking at, for example, a neurite outgrowth assay, 3D spheroids, organoids, and whole organisms such as Drosophila, zebrafish, and C. elegans. So you can work with various kinds of samples, both live and fixed cells. tissue slices. You can go up to the high magnification, up to 100x. So you are able to look at microorganisms and looking at unstained cells. So if you're doing imaging now and you are wondering, could I automate this process so that I can step out of the dark room and go, in fact, these systems don't even have to be in a dark room, but again, thinking about freeing up your time to be able to focus on other kinds of applications because these can be automated. And as I mentioned, for live or fixed cells. So just to kind of show you a little bit of example of a few different assays here, we're looking at some cells undergoing migration. We're looking at some cell division. We're looking at some engulfment of bacteria. And I'll go through a couple of these in a little bit more detail. Now, as mentioned, this can be for 2D or for 3D applications. So you can look at assays such as a 2D assay where you're looking at a single plane. You can take Z series, so you can do a 3D acquisition. You can look at that over time, and you can analyze projections or the full 3D volumetric data. Here we're seeing an example with a spheroid where we can slice through the spheroid, and we can, within the same software, you can do the projections, you can do these rotations, you can make these movies. So it is a complete solution for that workflow. Now, you can also work with various kinds of formats, as I mentioned, slides or multi-well plates. And in addition, we have seen a really big growth in the area of these 3D optimized formats. So I wanted to just share an example of a few of those. We're looking here at a micro well. This is what you're seeing here is the well, but it's actually micro wells where you have in these plates a spheroid growing in each of the micro wells. So it's a micro pattern here. Round bottom plates, U bottom plates, very popular in working with spheroids. So you get one spheroid per well. You see one forming here. 3D optimized plates with regard to having hydrogels and cross-linking. So here you see a nice matrix of neurons growing in this 3D hydrogel format with the cross-linking. And on the right here, you see an example of the Mimitas organ-on-a-chip microfluidics plate, where we're looking at various types of disease models in this microfluidics environment. And what you'll see as I go through the presentation is the ability to work with various kinds of formats, such as the examples I show here, really has to do with the ability to automatically focus on these formats and acquire data in 3 dimensions. You can look at stained or unstained cells. You see a few different examples here. This is a combination of wide field and confocal imaging. You see it's a micropattern spheroid, cardiomyocytes beating. So these are stem cell derived cardiomyocytes being imaged in transmitted light. And you can even do your colorimetric imaging. So let's take a look now at an example of 2D and then some 3D assays. Now, if you are new to automated imaging, you might be wondering, how are you going to handle visualization, analysis, and so on of larger data sets? If you're used to maybe looking at a couple wells, being able to have this data done in an automated way, well, there's tools for that. This is a montage view of the entire experiment. So this is the whole plate that you're looking at. This is a compound screen. And it's very easy to see those trends. So the visualization here on the image side, you could see, for example, if you were having a if you have dose response, you can see your controls and so on. This is interactive, so you can click and look at the full resolution images. Now here you see an example of one of those wells, 2 of those wells. We have our control. This is in wide field. This is an example of a 2D wide field location. On the left, you see the control. So here we have the nuclei labeled, we have a target protein labeled, and we have EDU. So we're looking at a viability type of assay here. And on the right, you see an example of a compound treated. Right, so in this case, we are looking at, you can see an increase in the expression here of the target protein. And as I mentioned, that workflow, acquisition, analysis, in data visualization, here you can see the software segmentation. This is the analysis. So the software can identify, right? So you don't have to think about manually clicking on cells or drawing circles or so on, or manual counting. The software can identify, in this case, you can see the positive nuclei, positive for that marker, and the negative nuclei here in gray. So from this, we can get measurements such as intensity, area, shape, and so on. So that right within the software, you're able to see that data. In this case, you're looking at the heat maps of various measurements. In this case, we're looking at how many cells were positive for the EDU. So you can immediately see the trends here across the plate as we saw in the images. Total number of cells, intensity, these are just a few measurements. There's, can be 10, 20, 30, 40 different measurements depending on, and more, depending on your analysis. So the data is all integrated into a database. You can export it to Excel depending on your downstream workflows. Here's another example. This is a receptor internalization assay. Again, another example of a 2D assay that I'm sharing here. These are U2OS cells that stably express a GFP beta arrestin. And so in the unstimulated state, here we see the nuclei. In the unstimulated state, we have GFP everywhere on the cell, as you can see some of here. And when treated with compounds, in this case, the positive control is treatment with isobutanol, we get receptor internalization, and then we see the formation of vesicles or puncta. This plate montage view is interactive. So just clicking on the boxes brings up that full resolution image. So working with the data is quite easy because it's designed the software around working with multi-well plates and slide formats. So here you can see the nuclei on the right, and now we see at this intermediate concentration, we see the formation of these vesicles. And as we move across in the dose response here, we can see we get bigger and brighter vesicles and so on. So the software can automatically identify those and then measure, for example, in this TransFloor GPCR assay that you just saw, we can then take those results, we can do curve fitting and get statistics on that, such as the ECT data. So as I mentioned, we can work with live cells also. Here's coming back to one of those live examples. This is an apoptosis assay. So we're looking at phase contrast and transmitted light in gray, as you see, the nuclei in blue, caspase indicator in green, and a dead cell diethidium dimer in red. This is being imaged every 10 minutes with temperature, CO2, and humidity control, and that's over 16 hours. You're looking at one well, but this was done across the whole plate in an automated way. We then wanted to determine how many cells are present, how many are live, how many are dead, how many are early or late apoptotic. And that's what you see here in an example. So on the left, you're seeing the data overlaid, and on the right, you're seeing the segmentation result. So the color coding here represents the different population. We have our viable cells, we have our apoptotic cells, we have our early apoptotic, and then this is based on the presence or absence of the different markers, late apoptotic, and our necrotic. And so here you can see with our control, as well as our negative control and our positive control here, as well as two different compounds, that we're then able to look at that over time and measure the number of cells in each of those different populations. So we can see clearly when we are inducing apoptosis, we see an increase in those late apoptotic cells, and then depending on the compound, we may or may not see that increase also. So those were just a couple examples with some viability, internalization, as well as apoptosis, examples of some 2D assays. Now I'm going to move into looking at some 3D assays, and this is definitely an area that there's been a lot of growth over the last several years, and we are seeing more and more customers interested in moving into these 3D assays, as well as more options, more formats available with regard to different optimize 3D, plate types as well as sample formats and so on. Here you see an example of stepping through in Z of a spheroid where we're looking at markers here for viability as well as markers for different cell structures. And on the right, you're looking at the projection. So that is taking the Z series and then showing the projection in a single plane. So a 2D view of the 3D data. And this can all be done within the software. So in that typical 3D cell culture workflow with spheroids, as an example here, so culturing those spheroids, as I mentioned, this can be done in different 3D optimized formats. Here you see an example in a round bottom plate. And those can be cultured directly in the ultra low attachment plates, as you see here as an example, or they can be transferred depending on your assay setup. Then in the typical workflow, those spheroids are forming and then being treated with compounds for various time periods. Typically, that's 1 to several days, depending on the kind of mechanism that's being studied. Then stained for different markers. Again, this could be looking at viability markers. This could be looking at certain cytoskeletal elements and so on. And then acquiring those spheroids. So this is acquiring 3D images, and this is a typical workflow for spheroids as well as for organoids. And then automatically, again, being able to look at those results and then analyze, and I'll show you some examples of how you can look at 3D volumetric type of analysis. So in addition to acquiring the 3D data, actually getting volumetric 3D analysis. So here's that workflow I'm looking at a little bit differently here. So this is with that example that I showed, and we'll look at some of this analysis. With the formation of the spheroids, these are colon cancer spheroids. These are growing 2 days, and we're imaging at every 3 hours, so you can actually watch the spheroid forming. This is a round bottom plate. You can kind of see here on the outside in this view, that's showing you can kind of tell that it's round bottom, staining the cells and then imaging. Now, you can acquire 3D data and then make a projection to do a 2D analysis, or you can take that whole volume, the 3D data, the 3D Z-stacks, and do 3D measurements. Here are some examples of the types of measurements. In a spheroid example, we could be looking at the area, the volume, the shape, the perimeter, the number of cells. That could be either on the projection or 3D cell counting. The distance between the cells, we could be looking at, fiber, 3D neurite, for example, and I'll show an example of some other applications coming up. So we'll step through this example first. This is with the round bottom plate. So you're getting that spheroid formation. This was a toxicity screen of these tumor spheroids. And here you see the result, again, that plate overview montage where you can see all of the results quickly. And here you can see in this example assay, which is in the 384 well plate, we have increasing concentration from the bottom up in this plate. We have our controls here at the bottom. And then we're looking at screening different compounds to see the viability here and the effect on the spheroids to be a measurement of the viability, how they change the shape, the number of cells, and so on for these different compounds, with each having different mechanisms. So as I mentioned, this is an interactive montage. So you click on the areas and you see the full resolution images. So immediately we can see that these spheroids do look different. So this case that we're looking at right now, this was a 3D acquisition with a 2D projection. And then we went ahead and looked at the segmentation, so identifying each of the spheroids, and then the number of live or dead cells in that, okay, by looking at the different, in this case, the viability markers for the different compound treatments. So we can see, we've got the spheroids much smaller here in the saurosporium treatment. With the Taxol treated, we've got, a sort of losing the compaction, and with the topicide, we also see that combination there. So what we can get from this is cell counts, as well as looking at the morphology, intensities, and so on. Here you see those results, And we're looking at the number of cells per spheroid, or those normalized values. And what you can see here is, depending on the compound, we're certainly having an impact here in the total number of cells, how many cells are positive for our viability marker, and so on. Positive for our, and then negative here for having the ethidium dimer. So here you can see we're impacting the area of the spheroid, the number of live cells, the number of dead cells, and so on. Now, when moving into 3D types of assays, especially with spheroids, organoids, or whole organisms, or where you might have rare events, one of the challenges is actually finding those objects. So I want to step you through a workflow that we have called a quick ID and targeted imaging. And what this enables you to do, as you can imagine, let's say you were not working with a round bottom plate, or you had a flat bottom plate and you had one spheroid, or you had certain kinds of spheroids, or perhaps you're looking at cells and you want to find the cells that were in mitosis, or so on. What you can do is actually image at low magnification, find those objects of interest, and then automatically go to high magnification. So this is an example of finding targets and then moving into the higher magnification to do your imaging. This is faster, of course, than imaging an entire well at high magnification, less images to store, faster to analyze, and you're zooming right in on your data. Here's another example, right, in this targeted imaging. You can see that in this case, we're doing a whole well image at low magnification. In transmitted light, we're finding our spheroid. You can see their different positions. In this case, this is done with our water immersion with this thicker object. We can then automatically zoom in and acquire that at high magnification. As you see here, we went from low to 20x, and then do a 3D segmentation to look at individual cell counts, spheroid volume, and so on. Here's another example of that. This is using that quick ID or targeted imaging. And again, looking at low magnification, finding, in this case, you've got multiple targets of interest here. And this whole process is, much faster than just automatically going straight to high magnification. This is using the Symphony and VersaGel. It's a cross-linkable hydrogel matrix that's used to grow these spheroids in 3D space. And so this is imaging 2x and then automatically moving in and zooming in and imaging at high magnification, in this case, moving in from 2x to the 10x. So this is a cross-linking, cross-linkable gel that's forming these 3D spheroids. And we have more information, application notes on these different applications that we're happy to share with you if you are interested to learn more about how the assay is set up and so on. So please just let us know if there's something specific that you're interested in. We're happy to share those application notes with you. It's hard to encompass all those hundreds of applications in our discussion today. So let's look at some of the data here. So what you're looking at here is an early tox live dead assay. This is compatible with those spheroids grown in the VersaGel. And so what you can see is that these are treated with compounds for 48 hours, and you can see the differences here in the formation. And what we can then look at is at the different compounds and look at the proportion of live and dead cells. So again, taking this assay and automatically finding the spheroids by low mag and moving to high magnification in a targeted approach, and then doing an analysis for the viability markers. Another example of a 3D assay I want to mention is looking at cardiomyocytes. And we'll look at cardiospheroids. So first I'm going to show you the assay in 2D. This is an assay where we're looking at calcium as the marker, treating with different compounds, and then looking at the changes here in the beat profile. So on the bottom, you're looking at an example where we have our control, then we have different compounds, and what you're able to do is measure that beat profile. So these complex patterns, we can measure the spacing, the amplitude, and so on. Now this is a 2D example that you're seeing here. We moved this into a 3D cardiospheroids, whoops. And what you're seeing is these cardiomyocytes in 2D, then culturing that in 3D, and being able to see, so the changes that you're seeing in intensity are changes in the calcium. So we're looking at those calcium flux changes, and then do an intensity measurement. Here you can see with some of the different compounds that were treated, either slowing down or speeding up that beat rate. We can then make those measurements, and this can be done very fast. So here you see an example where treated with epinephrine, and on the bottom what you're seeing is this is a total of 2 seconds of imaging. So very fast frame rates are possible. So if you are wondering, you're looking at a very fast reaction, or you're looking at cells that are moving very fast, or in this case you see the calcium changes, that can be captured in an automated way across the plate. So we see control, epinephrine, and cisapride, and as you can see, we've got really speeding up that beat rate, so the intensity fluctuations, and cisapride slowing down. And you can look at this at over time here, and we can then measure and quantitate, again, those beat profiles, the spacing, the amplitude, and so on. The next assay I want to touch on is a 3D angiogenesis assay. In this example that I'm showing you, this is using the 3D angiogenesis model that's established by Mimitas, using the Mimitas organoplate 3 lane. And this is showing you an example here where you see it kind of flipped over, and you can see the chip, the pattern here. And what we're doing in this particular assay, again, we have a really nice application note to share more details with you on this. But what I'm going to focus here on is, in this angiogenesis assay, we've got the way that this organ on a chip is set up is we've got these 3 different channels, and we're seeing we've got our endothelial cells here on the top. So we're zooming in on one of these areas right here, and we've got collagen in the middle, and at the bottom is the proangiogenic factors. So what happens is you'll get a vessel formation, so a 3D vessel formation here, and then what we're looking to see is, are these proangiogenic factors going to cause the sprouting and the formation of the angiogenesis, the angiogenic environment. And what you can see here, and I'll show you a 3D assay, 3D analysis of looking at this, is what we're able to look at right now. What you're seeing is the projection. So I'll show you a rotation here of the segmentation coming up. Okay, so these angiogenic sprouts are allowed to form for up to 4 days, and then we're looking at them and fixed and comparing them depending on the different treatments. Okay, so you're getting a endothelial vessel formation here on the top forming. So this is all happening, and this is a 3D. So we're looking at the segmentation. So what I want to share with you here, so you're seeing the tube formation, the sprouts going down towards those angiogenic factors, that we're able to segment and identify that tubular structure. The color coding here represents the different tubes in the environment. Here you can see a nice rotation of that. So we're looking at the individual cells here in the tubule formation. And from this, we can then look at how many sprouts, what were the volume, how many nuclei, and so on. So this is a full 3D volumetric analysis in this 3D angiogenesis model. And we see some results here. So looking at across, as I mentioned, between 0 and 4 days, you're allowed to form. We're looking at the number of sprouts, the volume, again, it's a 3-dimensional assay, and then the number of nuclei in 3D. So for the next part of the presentation, again, we have example application notes, posters, and so on. If you'd like to learn more about any of these particular applications, as well as, of course, additional ones, what I want to do now in the next few minutes is just touch on a little bit more about the image express systems. So as I mentioned, this is a family of imaging solutions. We've got wide field imagers, we've got confocal, we've got laser-based, we've got LED-based, and again, the software being an integral part of, as you saw, for both 2D and 3D assays. Within the software, there's tools for acquisition analysis wizards. You can see some examples here of the kinds of modules that are available for your assays, really, again, addressing a wide range of different application protocols. This is a little analysis module builder. You can see the data visualization. We've got, individual correlation here, individual measurements, being able to view the data in scatter plots, bar charts, and so on. So the data visualization, you have the heat maps. Again, especially if you are doing manual microscopy now and moving into automated, what I'm hoping to show you here is how you don't have to be concerned about how am I going to work with all the data. It is set up to work with these both on the acquisition and analysis workflows. So being able to see the data all at once here, like in a montage, in a heat map, in a bar chart, those are built right into the software so that you can think about being able to now interrogate the data more deeply because you've got all of this data connected together. So you can look at plots, we can get cell-by-cell data, scatter plots, individual segmentation, and then taking that data and then even working with multiple plates at once. So if you're doing a compound screen, you've got multiple plates combining all that data into a data mining approach. And here's a couple examples for you to see with some of those application module tools. The overlay here that you see is the image segmentation. Once from this, we can get multi-parametric data, cell-by-cell data, as well as summary. So here you see example of venereal outgrowth. A granularity, this could be any kind of puncta, this could be vesicles, DNA damage, internalization, and so on. Within the software, you can make those movies and rotations, so you can not only acquire and analyze, you can have that visualization and make those movies that I showed you. In the 3D analysis module, here you see an example of that. Again, we're getting 3D cell counts, we're getting distance, and we're getting volumes. For time, I'm going to go through the next couple slides quickly. I want to make sure we have a few minutes for questions, but we will always make sure that we follow up with you on that if we have additional questions. So here are some of the ways you can look at the data, and if you're moving in 2D or 3D, you can look at projections of its 3D, you can look at the side views, so orthogonal views also, distance maps, and doing that 3D volumetric segmentation. In the next couple slides, I want to share with you some new options that we have with our latest offering of our system, where we've got some additional tools here, and that is doing some AI and doing machine learning and classification. Here you see a nice example of a cell painting assay. So what we have available is some additional machine learning segmentation and classification tools with the Encarta software. So we've got deep learning segmentation, especially for those complex type of segmentation analysis, both transmitted light as well as in fluorescence. Classification, so being able to automatically identify the different populations of data. You see a nice example here with determining where cells are in mitosis, for example, and also having additional volumetric tools. So really what I want to share with you here, and you can see a nice example of segmentation, is that workflow for acquisition to the image analysis. The first part of this, of course, is identifying the objects to measure. So you've got tools here with the software applications that I'm mentoring across the family for being able to identify and classify the different populations. And here you see an example of that with the cell painting assay, where this is a multiplex assay where you are looking at, for example, 5 different compartments. You need to image them, and you need to analyze and segment those individual compartments to get that multi-parametric data. Now, these systems can also be integrated into larger automation platforms for anybody who is doing higher throughput. I want to just give you a few examples. You can see our Pico imager here. You can see our confocal imager here, both integrated with multiple instruments and with robotics. Another example here, we can see 2 of our plate readers with our imager, and even larger platforms here for our customers, for example, that are doing full automated compound screening. You've got incubators, you've got liquid handling, and so on. So to summarize the systems, again, we didn't focus as much on the specifications. I just want to kind of summarize here. We have wide field and confocal imagers. You can do live cell. You can do transmitted light or phase contrast, fluorescence, 2D and 3D assays. This is an onboard pipetter for anyone doing fast kinetic assays that wanted pipette in the middle of their experiment. And depending on the system, all the way up to 100x again, to really be able to address a wide range of applications and assays in 2D and 3D formats. So with that, I want to thank you